Hey everyone, I hope this podcast and my blog and books have been helpful resources for you and will continue to be. But if you've been struggling with a chronic health problem and are feeling stuck, consider coming to work with my team and me at the California Center for Functional Medicine. We work with patients all over the US and have experience treating a wide range of conditions, including GI problems, autoimmunity, hypothyroidism, cognitive mood and behavioral issues, weight gain and metabolic dysfunction, and more. Our unique model teams clinicians with nurse practitioners and health coaches, all of whom are trained in my ADAPT framework approach to provide a high level of care to our patients. This means more support between appointments, personalized guidance on diet, lifestyle, and behavior change, a cutting-edge patient portal with 24/7 access to your labs and records, handouts and resources to guide your protocols, and a team of practitioners working together on your case. We're currently accepting new patients, so if you'd like to learn more, visit chriskresser.com/becomeapatient. Hey everyone, Chris Kresser here with a brief message before we begin the podcast. Imagine a career that draws on your passion for wellness and disease prevention, that harnesses your ability to support and connect with others, that helps you to develop as a person while you help others to do the same. What if you could earn a living while making an impact on thousands of people's lives and even on the future of healthcare? That'd be pretty great, right? Well, that job does exist. It's the job of a health coach, and I believe that health coaches will play a crucial role in the future of medicine, not only in the US, but around the world. 1 in 2 Americans now has a chronic disease, and 1 in 4 have multiple chronic diseases. Chronic disease is destroying our quality of life, shortening our lifespan, bankrupting governments, and threatening the health of future generations. And our medical model is not prepared to address it. Why? Because the only way to prevent or reverse chronic disease is by changing our diet, lifestyle, and behavior. And conventional medicine is simply not set up to do this. Bottom line, we need people who can provide this support to help people make the changes they need to make to save their lives. We need empathetic and compassionate people with a skill for connecting and a passion for change. And my job is to make sure they have all the training they need to do their job so well they will change the future of healthcare. A career as a health coach can be incredibly fulfilling both professionally and personally. It really could change your life, and it can also change the world. That's why I'm excited to announce that we're launching the ADAPT Health Coach Training Program. It's a 12-month, 100% online certification that will prepare you for a successful career as a health coach. It includes training in core coaching skills, functional health, ancestral diet and lifestyle, and professional development. And it's unlike any other health coach training program currently available. To be notified when enrollment opens and to learn more about the program, visit kresser.co/success. That's KRESSER.co/success. Now on to the show. Hey everybody, Chris Kresser here. Welcome to another episode of Revolution Health Radio. This week I'm really excited to welcome Dr. Tommy Woods as my guest. Dr. Wood is a research assistant professor in the Department of Pediatrics at the University of Washington, specializing in neonatal and pediatric brain injury as well as long-term brain health. He has an undergraduate degree in biochemistry from the University of Cambridge, a medical degree from the University of Oxford, and a PhD in physiology and neuroscience from the University of Oslo. Tommy previously spent 3 years as the Chief Scientific Officer of Nourish, Balance, Thrive, using advanced biochemical testing to optimize health and performance in athletes, as well as developing software tools to help understand and interpret blood tests. Alongside his main academic research, he continues to work with elite athletes, as well as developing cost-effective and easily accessible tools to help people improve their health and reverse chronic disease. I'm really excited to talk with Dr. Wood about consumer genetic testing. I'm sure many of you are familiar with these tests, like 23andMe, and there's a lot of awareness now in our field about single nucleotide polymorphisms like MTHFR or COMT or FTO, and many people are making decisions about how they eat and what supplements they take based on polymorphisms in these genes. Dr. Wood has done some very interesting research in this area, and I think the results may surprise many of you. So, without further ado, let's dive in. Tommy, welcome to the show. I'm so glad to have you. I've been looking forward to this.
Yeah, me too. Thanks so much for having me.
So we're really going to dive into this topic of consumer genetic testing and what useful, actionable information that we can get out of it, both as consumers, for lack of a better term, just people who are interested in improving their health, and as practitioners who are working with patients or clients using this data or these data. So maybe we can start just from the 30,000-foot view for the listeners that are not as familiar with some of the basic terms that we might be using in this interview. What is a genotype? What is a phenotype? You talk a little bit about just the big picture differences between genetics and epigenetics to set the stage.
Sure. So genotype, depending on who you're talking to, might be used to mean a few different things. So in general, in its loosest term, it basically means your whole genetic sequence in a given person. So people who are familiar with some of the biochemistry will know this is about 3 billion base pairs. These are nucleotide bases. They may have heard of A's and C's and G's and T's. This is basically the code within your DNA that is then used to make up certain genes, of which we have 20 something thousand. Each one is made up of hundreds to potentially millions of these bases, and they code for a protein which does something in a cell. When we're then talking about genotype and disease risk, we're most frequently talking about potential polymorphisms, so changes within the code of a single person relative to other people. So you'll hear something like single nucleotide polymorphisms or SNPs. And this is basically where a single base, a single one of those A's, C's, T's, and G's is changed. So say from a C to a T, and this might then alter the amino acid that is made in the protein. It might somehow alter the function of the gene or the protein. And for each of these SNPs, you can be one of three things. You can either have 0, 1, or 2 copies of that SNP. So you can be homozygous. That means that you have 2 copies of the same, or you could be heterozygous, which means you have 1 copy of each. So in that setting, when we're talking about genotype, it's usually your genotype for a given SNP, or maybe a collection of SNPs that all affect a certain phenotype. A phenotype is a measurable characteristic, which we usually think of as being the interaction between your genetics and the environment. So this can be anything that you might measure as a practitioner or as a person, as you said. And this so it could be heart rate, blood glucose, your height, your weight, your homocysteine level. If you're worried about methylation, we'll talk about that later. So anything you can measure, that's usually part of the phenotype. When we talk about genetics, this is generally just the study of the heritability of genes. So like how much is a certain phenotype heritable or associated with a given genotype, or how much is a disease risk associated with a certain genotype. When we talk about epigenetics, these are the factors that are associated with changes in the expression of genes rather than changing the genome itself. And this happens at multiple different levels in the cell, but people may have heard about things like methylation. You can methylate the DNA itself, add a methyl group to the DNA, or acetylation. You can acetylate the histones, which are the proteins that DNA is kind of wrapped around. And this changes the way a gene might be accessed in terms of its ability to be turned into mRNA to then code for the protein, or various different ways that you might change the function of that gene. And the epigenetics determines how we express certain genes, but it also determines the actual cell itself, right? Every cell has the same DNA, but different genes will be switched on to make a muscle cell versus a brain cell, for instance. And epigenetics is very hot at the moment. You'll often hear people talk about, it's not the genetics, it's the epigenetics. And that is certainly true. And we can definitely talk about the broad strokes of epigenetics. That's not really what we're going to talk about today, but we know that there are certain methylation patterns you can see as like a person or a cell ages. There are certain factors that we know are directly associated with a certain methylation pattern. So you can see certain patterns of people who smoke, and if they stop smoking, some of that pattern will come away. But the study of epigenetics is kind of like the next frontier, is incredibly hard. Like, people who are talking about epigenetics may not realize that it's incredibly expensive, it's incredibly complex to do, and we still don't really know how a given environmental input changes the epigenome of a given cell. And just as an example, there's a nice paper that came out recently, and I'm a neuroscientist. I spent most of my time looking at the brain in the lab. And a recent paper came out that said that in a mouse, there are 565 transcriptionally distinct cells in the mouse brain. And what that means is, if you look at the mouse brain, there are 565 different epigenomes, or cells with different types of epigenome in the mouse brain. I mean, so already talking about hundreds of cells just in a mouse brain, which is much less complicated than the human brain.
We're going to need AI.
Yeah, and then to think about how each individual environmental component is going to change the individual epigenome in every cell. Like, we're talking about millions, billions, trillions of data points, and looking at how they interact is, we're a long way away from figuring out how that's going to work.
Not even close. Boggles the mind. It's one thing for AI to beat someone in Go. It's another thing for them to decode the epigenome. It's
Absolutely.
a whole other level of intelligence.
Yeah.
So one of the things you mentioned genetic changes are heritable, and one of the biggest discoveries in epigenetics was that those changes can also be passed on to future generations, which is important, I think, to remind the listeners, because that was a previous misconception that only our genes could be passed down, but we now know that even epigenetic changes can be passed down to at least one successive generation, but a lot of studies are showing it could be even 2 or 3 successive generations, which means, of course, that the choices that we make don't just impact us, they actually impact our future generations if we're going to have children and grandchildren. So that's a great intro and a good starting place. And let's begin with, what the positive aspects of this kind of testing are, like what the potential value is of knowing our genes. What kind of outcomes can we imagine in the future once we do get our heads around this stuff, or AI does that for us? There was obviously a reason that we started going down this path. So what do you see as the potential for this once we're able to really realize that?
Yeah. So the original hope of the Human Genome Project, and since then we're doing what we call genome-wide association studies, where we look at individual polymorphisms and a phenotype or a disease risk it might be associated with. But the hope is that we can identify people who are at high risk of a given disease. We can discover or target specific pathways, and give personalized therapies based on a person's genetics. And at the moment, this is kind of something that is suggested in the direct-to-consumer market, but it's really not something that we're able to do. So there are an increasing number of academic works in the published literature where they take what we call now polygenic risk scores. So this might be dozens or hundreds even of SNPs, and then look at, stratify people based on their then subsequent risk of something like Alzheimer's disease or cardiovascular disease. And then within those subgroups, you may want to give, a different type of intervention. So when one study has looked at people's responses to statins in those who are at risk of cardiovascular disease, and in those with the highest genetic risk, they seem to respond best to statin therapy. So you might be able to identify one particular subgroup that's really at risk, and they might respond better to a therapy. Whether statins are the approach that we wish to take is a whole different conversation that I don't think we're going to have here. But one thing that I still, if you want to step back a little bit from that, one thing that is kind of a common theme in almost everything that I've seen in the world of genetics is that I'm still not really sure that when you identify somebody at genetic risk, you're going to change what you're actually going to do with them. So again, if we stick with cardiovascular disease, there was a paper in the New England Journal in 2016 called Genetic Risk, Adherence to a Healthy Lifestyle and Coronary Disease. And they took 55,000 people, and they looked at 50 risk SNPs for coronary disease. And they found that those who had at least 3 of 4 healthy lifestyle factors were at significantly lower risk of coronary artery disease. And the effect in terms of improvement was biggest in those at the biggest genetic risk. But when you look at the things that they considered the healthy lifestyle factors, it was 3 or 4. It was not smoking, not being obese, physical activity at least once a week, and then a healthy diet pattern, which, they talk about red meat and those kinds of things, but it's basically a whole foods type
Yeah.
real food diet.
Yeah, we're not talking about like intermittent fasting and meditating for an hour a day, or like, cyclical ketogenic diet or sauna use or pulsed
no.
electromagnetic field therapy. We're talking about just the absolute basics, like not being the furthest down the track of the standard American diet and lifestyle, right?
Yeah, exactly. And the other thing to note is that the effect was, or these factors were beneficial regardless of your actual genetic risk. So if you're somebody who's worried about your risk of heart disease, don't smoke, try to avoid being obese, exercise at least once a week, and eat real food. I mean, but you would have told that to everybody regardless of whether they had genetic risk or not. So
exactly.
So even though we can start to stratify based on risk and potential interventions, I think for most of the people who are listening to this podcast, the genetics really aren't going to come into play because you're still going to recommend the same things.
Yeah, and I give a presentation on this topic. It's called "Why Genes Aren't Your Destiny," and I have a slide in there about a study. You probably saw it. It was, I think, 2018, and it was a large study, and they found that adopting just 5 health habits extended lifespan by an average of 12 years in men and 14 years in women. So it was 13 years on average. And those habits were healthy BMI, at least 30 minutes of moderate activity a day, not smoking, not drinking excessively, and following I'm doing air quotes here, healthy diet. Same thing there, right? So again, not like super advanced stuff here, just following your basic healthy lifestyle factors, and that increased lifespan by 13 years. I mean, that's an enormous difference. And I'm often reminding people, as I know you are, Tommy, like a lot of the studies that you hear about on the media are not about you meaning if someone's listening to this podcast and they're doing even those basic things, you can't make the assumption that these data in the studies have really anything to do with them.
Absolutely, yeah.
So we had this promise, and it was, world enough to remember all the exciting predictions from the 90s about how decoding our genome was going to completely, revolutionize healthcare and change our destiny. And another slide I have from that presentation is from Craig Venter, who ironically was one of the first people. He was one of the first to sequence the genome, but he was also one of the first to recognize its limitations. He said, "We simply don't have enough genes for this idea of biological determinism to work."
Yeah.
Which is kind of related to what you're saying. Like it's really more directed to the environment. So let's talk more about that. So we had this decoding of the human genome, this exciting promise that if we understand our genes, we're going to be able to, revolutionize healthcare. There is some significant potential value somewhere down the road once we have AI that's sophisticated enough to really work through this. But even then, as you pointed out, is that knowledge really going to alter the recommendations that we would make on a public health level? And also, if you're a practitioner working directly with patients. So those are the big questions. Let's dive in now a little bit to the more specifics here. I know you gave a great talk at AHS. I unfortunately wasn't able to be there, but you generously sent it to me, and I watched it and really enjoyed it. And you talk a little bit about some of the currently available consumer genetic tests and also the algorithms that you can put your test results through, like 23andMe and MTHFR support. And not to single any algorithms out here, because I think most of these are from people that I know we both respect and are doing good work and are out there trying to help people. But the question is, when you really dive deeply into this, how much value do we get out of the information that comes from this? And I guess the corollary question would be, is there a risk in putting too much attention and energy into this kind of information? I definitely have some thoughts about that from my experience as a practitioner, as I imagine you do. So, let's talk a little bit about these tests and what they can tell us and not tell us.
Yeah. So we talked briefly about the state of the academic literature, and I think that there is potential promise there, but none of that really has been translated to the consumer market yet. And when you read the academic genetic literature, they're very clear about the fact that individual common SNPs that you'll get on a 23andMe, or if you run it through some third-party tool, that they tell you almost nothing about your individual disease risk. Yet, when you're presented with your data from whoever it is you've maybe paid a few dollars to analyze your 23 or ancestry DNA report to give you some idea of risk, they're saying that this SNP is associated with X% increase in the risk of diabetes. And in reality, the population data is so variable that it's almost impossible to determine for an individual, how much their risk might be increased or decreased. And this is the problem with translating these large studies. Now, they're doing genome-wide association studies with hundreds of thousands of people looking at, hundreds, if not more, SNPs. And you can definitely stratify that whole population based on their risk. But, for an individual, the absolute variability in their potential outcome is so large that it's almost impossible to tell them about their risk. And the problem is, or one of the biggest problems that I see, is that when you read a paper, what they give you what we call summary statistics. So they'll tell you like the average effect of a genotype. And then they'll give you an error, something like a 95% confidence interval, or the standard error of the mean. And it gives you these nice tight little error bars on a graph, which makes them look really good. And you think, okay, wow, this, if I have this SNP,
this is a real strong signal here.
This is a real strong signal, absolutely.
Yeah.
But what I did in the talk, and what I've tried to do some of since, is if you have that information, you have the average, and you have some kind of confidence interval, you can recreate a whole bell curve of potential effect, and you can really start to see how variable some of this data is, and then you can start to calculate the actual effect of having a certain amount of a certain SNP. But when you're looking at what we have currently in the consumer market, they're very much still focusing on these individual SNPs, which the academic genetic literature left a decade ago because they appreciated that it was almost useless. And sometimes they'll maybe collect together like a handful of SNPs that are associated with a given phenotype, say being obese, but they haven't like weighted and accounted for the error and how these things combine, like you would have to do if you were doing a proper study that you were going to publish. So again, like every time this is presented to you as a consumer, the effect of a given SNP is hugely overestimated. And then when you think about how people are applying this, now I see adverts all the time for do this genetic test and we'll tell you how you can eat or should eat. Do this genetic test and we'll tell you about your response to exercise. I work with a lot of athletes, so I've had to debunk some of these genes associated with athletic ability in a similar way as some of the health related ones, and it's exactly the same. The response to a given training program is hugely variable, and your genotype is just a tiny portion of that. So I mean, they're basically promising things they can do with your genetic results that is basically impossible. And that doesn't even include the fact that these tests can be quite inaccurate. And I know you've written previously about the fact that there was a study where they compared different genetic tests from different companies, and they didn't all agree on SNPs in specific genes. And it even gets more serious than that. Occasionally, a mutual friend of ours, Chris, I know she had her 23andMe tested, and then they highlighted that she had a twin in Germany or something like that, and she wrote back and and was like, "This is physically impossible. I know I don't have a twin in Germany," and they'd sent her the wrong results. Like all of her SNPs were wrong. And she only realized that because of this twin association. So like when you're trying to make these decisions on something, that even if the data is it's not really associated with the outcome in a meaningful way, or it's so highly flawed, or there's errors in the process, like with any medical test, you really just don't know what you're doing.
Mm-hmm. Yeah, that's pretty scary. I want to touch briefly on something that you covered in your talk, which is relevant to what you just said. So Robert Sapolsky quote from Behave, where he says having the warrior gene, which maybe we'll talk about later, probably has less of an effect on your behavior than does believing that you have it.
Absolutely.
And then there was a study a while back, and I imagine you saw it. Maybe you can refresh my memory on the particulars, but essentially they took a group of people, they separated, randomized them into two groups, and told one group that they had SNPs that affected their exercise
Hmm.
performance in a negative way. And then they didn't tell the other group that. And then the group that they told that they had those SNPs, they actually did perform worse than
Yeah,
the other group, even though there were actually no differences in their in their genotype.
yeah.
That's the nocebo effect, right? Which we know is very real.
Yeah, absolutely. So this is Turnwald et al.'s in Nature Human Behavior this year. Yeah, absolutely. And they randomized people to being told they had a certain genotype, regardless of what their actual genotype was. And those who were told they had the good endurance genotype, I'm using air quotes here.
Yeah, yeah.
They had then when they redid a treadmill test, they sort of performed about the same. But those who were told that they had the bad endurance gene ended up doing worse. And they also did something similar with FTO genotype, which is the the
Yeah, we're
SNP
going to
most
talk about
associated,
that.
which we'll talk about. And they first gave people a test meal and then looked at measured hormone levels, things like GLP-1, and then looked at and asked about their satiety. And they did the same thing, and they told the people who they told had actually the protective version of FTO. This is one of the few examples where you actually might get a placebo effect from genotype. When they told them they had the protective version of FTO that was associated with less risk of obesity, when they repeated the test meal, they had higher levels of GLP-1 and higher levels of satiety, again, regardless of their actual genotype. So being told about your genotype or thinking you have a certain genotype actually changes physiology. It changes the hormones you can measure in your blood. Like, it's just incredible the
Yeah.
effect that you can have by telling people about their genotype. And what worries me particularly is the way that we talk about genetics. So like MTHFR, again, we'll talk about it. Incredibly, mutations are incredibly common, and it's incredibly common to talk about MTHFR SNPs in the functional medicine world. And it always comes with language like, if your MTHFR isn't working properly, right? It's always
Right.
negative language. It's always saying, oh yes, you have a deficiency. Oh, your MTHFR, is bad or it's
Yeah.
defective. And in reality, you're probably having a much larger effect on that person by telling them that than the actual gene itself.
Right. And that's why I asked the question, which admittedly was a little bit leading, is there potential harm here? I absolutely think that there is, because of what we're talking about now, both placebo and nocebo. I mean, there can be potential benefit, I suppose, if you're
Mhm.
trying to leverage placebo, like in the case of FTO, but much more potential for harm, because usually the way these are being used is to tell people how much higher their increased risk of disease is, not to tell them that things are going, you're in a great spot because you've got these phenomenal genes. It's usually telling people how much higher, why they're going to die sooner, or what kind of disease they're going to get. And I've had patients come into my practice and tell me they have the MTHFR disease.
Oh wow.
Um, so that's a big belief system to take on. Say
Absolutely.
I've got the MTHFR disease, which means that I'm always going to have this issue and be struggling.
Yeah. Usually if I talk about MTHFR, I like to point out that only 15% of people have the, quote unquote, best version of MTHFR. So all of us, I mean, the vast majority have some kind of MTHFR mutation. It's just,
Right. It's
it's,
highly
it's,
prevalent, but
it's
has low penetrance, right? To use
exactly.
the
Yeah. Yeah.
genetic speak. And if a gene or if a SNP is, 35% of the population has it, we have to really wonder what the clinical impact of that is going to be.
Exactly.
Right. Well, let's talk about some specifics now, because we've mentioned a few that we were planning to discuss, which FTO and MTHFR, maybe COMT, if we have time for it. So you did some really interesting data analysis in your presentation, and I'd love for you to just go through some of these common SNPs that people talk about and get concerned about, and tell us, what you found in terms of the actual modification of risk that
Mm-hmm.
they pose or don't pose.
Yeah, so we can start with MTHFR, seeing as we were talking about that most recently, and it's actually a really nice case study of looking at how different SNPs affect the function of a protein, which then affects phenotype, because there are two common SNPs that we talk about in the MTHFR gene. They are A1298C, so A being replaced with a C, and then C677T. And those who are 1298AA and 677CC, those are the people who have quote unquote 100% MTHFR function. And the way that they measure that, just in case people are interested, is they take people's blood cells or a tissue sample, and then they extract out the protein. This is an enzyme, and then they test it in a test tube to look at like how well that protein works. And later on, particularly with COMT, is quite a nice example of how just like looking at that in isolation maybe doesn't really tell you that much about what's happening in the system as a whole. But anyway, if you combine these different SNPs in different combinations, you get a range of activity of MTHFR. So those who are 677TT, they've lost about 70%, 75% of function compared to the best version.
Yep, that's me.
And I am the next worst, which is I'm heterozygous for both. So I've lost about 52% of my function. And
that's a compound heterozygote.
Compound heterozygote, exactly.
Yeah.
And so what you can do is you can take all of these different activities, sort of average activities of the different SNPs, and then you can look at something that we might care about, like homocysteine level, which 5-MTHF, which is produced by MTHFR, is used to recycle homocysteine, as is choline, which is one of the reasons why people might recommend more choline for people with certain MTHFR SNPs. But if you look at the whole range of potential function, your MTHFR SNPs only explain about 1% of your homocysteine level. Like it's just it's so little that it's a rounding error on the lab
test. A lab error, yeah. I mean,
exactly.
That's inside of the standard variation you get from testing homocysteine every day.
Absolutely, yeah. And then if you sort of create these full bell curves that I talked about, you compare me with my 52% loss of function to the best possible person, the likelihood that I have a homocysteine that is elevated greater than a person with the best MTHFR function is just 8%. Right. So then more than 90% of people who have my specific genotype will have no change or no difference in their homocysteine relative to the people who have the quote unquote best MTHFR function. So you can see, like, overall, the effect is so small that it's barely even worth worrying about.
That's so interesting, just that specific number, because there was that big study that was, I can't remember if Alphabet had something to do with it, but they analyzed genes and they found that over 90% of what determines lifespan was environmental, not genetic. And then there was another, a lot of the studies that have been done on chronic disease risk show that about 85 to 90% of the risk of chronic disease is environmental and not genetic. And as I was going through your presentation, it seemed like most of the genes that you were talking about that fall into that 10% or less influence number, which is consistent with these larger studies that aren't looking at specific SNPs, but they're just looking at overall patterns.
Yeah, absolutely. You can, if you look at any particular SNP, even like compare best to worst, however you want to call it, like the actual likelihood that you'll see a meaningful effect is always less than 10%. And even when you start to compound these in large polygenic risk scores, that still only increases the risk incrementally. And that's when you're comparing like the bottom 1% to the top 1%. So you may have like a 20% increase in risk, but like the middle 98%, like your genetics really aren't going to tell you anything, and it's almost all of it is an expression of the environment itself. So when you're thinking about MTHFR SNPs, we know that there's some requirement for nutrients that are going to make the whole cycle run, and we can test those. We can test homocysteine itself, and you can intervene as a practitioner. You may want to, supplement with certain nutrients or make sure somebody's getting more of a certain nutrient in the diet. We might, for this whole thing, we might think about B12, riboflavin,
just
folate.
Eat liver.
Just eat liver. Exactly. This is the thing.
Which is what we've been saying for years. Right?
If people just ate liver and eggs, if they're not sensitive to eggs, like this whole problem goes away. You don't even need to bother measuring any of it.
Just save yourself the trouble of going through all this stuff. Yeah. No, I know, Alex Leaf has dug up some interesting info on homocysteine variability really kind of disappears when you consider riboflavin or a factor in
Yeah.
riboflavin status. So in other words, if riboflavin deficiency is really what explains most of the difference in homocysteine levels in people with MTHFR.
Yeah.
And we're
And
not
that
talking about huge amounts, right?
no,
Like,
it's 2 milligrams. I think they tested 1.6 milligrams in that study, and this
yeah,
is particularly in people like you who have the 677 TT. But either
right.
way, if you just took 2 milligrams of riboflavin, if you're not getting it from your diet for whatever reason, again, like the whole problem goes away. You don't
Problem
need to worry
solved.
about it.
Yeah.
Yeah.
Or at least problem that you think is being driven by MTHFR. So it
Yeah.
might not solve all your problems, but it,
Yeah.
problem solved from the perspective of the influence that your MTHFR polymorphism is having on your health.
Absolutely. Yeah.
Yeah. So I know this is going to be a head spinner for a lot of people listening because MTHFR has been really, I mean, I find I'm constantly having to kind of talk people off the ledge when it comes to MTHFR because, in my intake paperwork, like I said, I've had patients say they have the MTHFR disease. I have people the forms I use, I have people list their top 5 concerns, and it's not uncommon for people to list MTHFR in one of their top 5 concerns on the intake paperwork. So, we're always living and learning in healthcare, and I've certainly was not blameless in my early enthusiasm for MTHFR, and as practitioners, we're always looking for ways that we can help our patients. And
Absolutely.
there certainly were early data that suggested this might be a lever that we could
Mhm.
push that would lead to positive results. So all I can say is I'm sorry that
Oh
I
yeah, I
contributed to
did it
that.
too. I did it too. Yeah,
And
absolutely.
and the best we can do is continue to learn and be willing to change our mind in the face of new data, right?
Yeah.
I mean
Yeah,
that's
absolutely.
that's why they call it a practice. I'm constantly reminding myself of that and my patients as well. So yeah, then what about FTO? So this is a gene that it's gotten a lot of attention. You briefly mentioned it earlier, talking about obesity risk, and of
Mhm.
course now we have 7 out of 10 Americans who are overweight and 4 out of 10 who are obese. So this is obviously a big concern.
Yeah, absolutely. And FTO is or fat mass and obesity associated protein is like the protein or gene that's most strongly associated with the risk of obesity or being overweight. We're not really sure what it does, or in some tissues at least, but in the brain, it seems to be associated with regulating food intake, which kind of makes sense. And if you look at the various meta-analyses, it seems like for each copy of the particular, there are multiple different SNPs in FTO, but there's one particular one that is talked about the most. And for each copy you have, your BMI on average goes up by 0.3, and it's maybe about 2 pounds per copy. And so you think about that and you're like, okay, there's potentially something meaningful there. If I have this, and again, like for most of the things that I talk about, I'm either heterozygous or I have the worst version. It sounds like you're potentially the same, Chris.
Yeah,
Um,
I'm screwed.
and and so when you look at, so when I do a similar analysis, right, I sort of recreate a full bell curve and try and look at what the full possible range of results for a given person, rather than saying, okay, well, I have this SNP, so I'm definitely going to be 2 pounds heavier. And it turns out that your FTO genotype predicts about 0.2% of your BMI. I think in the talk I was like, that's basically whether you had a glass of water before you weighed yourself or not. And so even though it's the SNP most strongly associated with obesity, on its own, it's essentially meaningless. So over the years, there's been an increasing number of these polygenic risk scores that I talked about earlier, and obesity is a really nice example. So one that I talked about in the talk, I spent a long time analyzing. They looked at 8 SNPs, the 8 SNPs most associated with obesity, and both in the paper, and then when I tried to recreate the data set and analyze it myself separately, they agreed quite nicely. It was about 4 to 5% of your BMI is explained by these 8 SNPs that are most associated with obesity. And like I was talking about earlier, in this particular paper, if you compare the bottom 1% of risk to the top of 1% of genetic risk, there's just a 20% chance that those in the top risk profile will have a BMI that's greater than that in the bottom risk. or the bottom, risk group, bottom 1%, 1%, top 1% versus bottom 1%. And, that 20% like potentially sounds like a lot. But when you think about these just like right at the extremes of people, I think, and then in the middle, there's basically no effect of your genetics on your BMI, or the predictability is very, very low. And then, so that was only 8 SNPs. And more recently, I was looking at another paper that came out in Cell this year, and they looked at 141 SNPs associated with BMI. So we're trying to sort of add this up and increase our predictability. And one thing that really bothered me about this paper is that they only give you the average BMI in each genetic risk group. So I have no idea about the variability. And this is incredibly dishonest to do this in like from a statistical viewpoint, because you have no idea of knowing like what the actual overall pattern of risk is for each genetic group. I mean, I'm actually really surprised that Cell published it like that. But even then, with 141 SNPs predicting, associated with BMI, that only predicted 13% of the variability in BMI. And in those people in the lowest genetic risk group, when they applied this to another population to test it, and it was the same in that previous 8 SNP score that I was talking about, the lowest risk people had a BMI above 25. So at baseline, the people that they're studying these genetic risk scores in are overweight or worse. So when you're trying to apply this to yourself, again, as somebody listening to this, I mean, and of course you may be struggling with weight loss, but if you're looking at your genetic risk, almost all of it is in an environment which is already driving people to be overweight and obese or obese, and the best genetic risk, or the people with the lowest risk, are still already overweight. And this is definitely an expression of the environment, because when you look at even polygenic risk scores and you look at what might modify the risk. So polygenic risk scores for obesity, if you're doing just the recommended amount of exercise, so we're talking about like 45 minutes to an hour, not of intense work in the gym, of just walking, standing.
Could even just be standing at your, if you work in retail, standing up or walking around. If you're,
exactly,
that's
enough.
Tabata.
Anything. It doesn't need to be strenuous at all, but just moving for an hour a day.
5 Metcons a week.
No. And that's enough to eliminate almost all of your genetic risk of obesity, which is incredible. But it also tells you that it's not going to have to be really hard work. And similarly, when they looked at the FTO genotype, there was a nice paper that came out in PNAS a couple of years ago. They basically showed that FTO wasn't associated with obesity at all until after the Second World War. So these risk genotypes, they're completely a reflection of the change, in the Western lifestyle. So if you're, again, somebody listening to this and you're moving frequently, eating real food, getting some sleep, maybe doing something to increase your distress tolerance, all of that, regardless of your genetics, and you would recommend that for everybody, then the risk disappears.
What would be even more revealing is if there was a side-by-side comparison in these studies of like, okay, well, modifying even if we had the technology, let's say through CRISPR or something, eventually to modify our genes, here's what the potential benefit could be in terms of risk reduction, less than 10%. And that's the best case scenario, right?
Yeah,
but
absolutely.
Here's the risk reduction that could come from eating a nutrient-dense whole foods diet, exercising, increasing your non-exercise physical activity and doing exercise, which are two different but related benefits, distinct, sleeping for 8 hours a night versus 6 hours a night, which is what 35% of Americans are doing now. Managing your stress a little bit, we could just stop there. I mean, I have no idea what the actual percentage of risk reduction would be there, but I'm absolutely certain that it would be, in the tens of percent, and way higher than what you could get by modifying the genes.
There's another example that I gave looking at risk of type 2 diabetes, which is on the rise in the US. And so again, I have a SNP in my melatonin receptor 1B that increases my risk of type 2 diabetes by 67%. But if you go and look at hunter-gatherer populations, their risk of type 2 diabetes is basically 0%. So even if they had the worst possible genetics, any percent increase above 0% is still 0%. So, almost all of this is an expression of the environment that we're exposing ourselves to.
And we also have these, migration studies that show what happened
Yeah.
to hunter-gatherer populations, or even people who are not hunter-gatherers, but were just living on a more traditional diet and lifestyle, like the Okinawans. And then they moved to LA. And, they acquire diseases at the same rate or even a greater rate than people, like Western Europeans. And that really interesting research of Weston Price, when he showed side by side pictures in his book of what happens to the dental, the structure of the jaw and the teeth and the palate in just one generation, moving
Yeah.
from a traditional diet to a Western diet. So yeah, it's really revealing. I'm curious. I know you talked about Conti in the presentation. I'm curious if you've looked into ApoE and relationship with cardiovascular disease and particularly Alzheimer's, because that is a gene that gets a lot of attention, and particularly with Alzheimer's. People who are homozygous for ApoE4 are basically told, like, look, just start making your plans for having Alzheimer's, because you're going to get it. I mean, that's what patients are told. So
Yeah.
have you looked into that?
Yeah, briefly, and ApoE is an interesting one because it's probably the gene which has one of the highest individual heritability scores. It explains about 5% of the heritability of Alzheimer's disease, which is much more than any of the other genes that we've talked about. And yeah, you're right. That's kind of how we talk about ApoE4, but it's very interesting because there is enough data to suggest that it is also very dependent on the environment that we expose ourselves to. So in the setting of continuous neurological insults, ApoE4, or being either a heterozygote or a homozygote, that changes the way we respond to injuries to the brain, and that injury could be either being punched in the face if you're a boxer, or being in a car accident, or it could be massive glucose spikes because you're eating loads of refined carbohydrate. Those are also neurological stresses that ApoE4 may exacerbate the response to. So in that setting, ApoE4 certainly does seem to increase risk, but in a more sort of hunter-gatherer setting, they looked at this in the Bolivian Simone, and they showed that those who had a high parasite burden, and that was elevated eosinophils and/or some kind of evidence of a parasite infection, they actually had protection
They
of
were
cognitive
protected.
function.
Yeah.
So yeah, in a Western in the Western world, if you are an ApoE4 carrier doing Western world type things, yes, I do think that ApoE4 is something that's going to contribute to your long-term Alzheimer's disease risk. But if you're doing all the things that we might recommend, again, to decrease the risk of Alzheimer's, and that diet, sleep, stress, we've talked about all of those already. We actually have very little evidence to suggest that ApoE4 is going to be that much worse than any of the other ApoE genotypes. And again, this is kind of just conjecture at that point, but all the other data would suggest that you're probably doing most of the things that are going to at least dramatically reduce your risk, and that is modifiable with if you are an ApoE4 carrier.
Right. And when I looked into this several years ago, I came to basically the same conclusion, that it affects our response to oxidative stress and inflammation in the
Yeah.
brain. And so if you want to take steps to reduce your risk of Alzheimer's, reduce oxidative stress and inflammation, which is, as you pointed out, the same thing you should be doing even if you don't have that genetic risk for Alzheimer's. That's still the name of the game, basically. But this is the one that I get most concerned with in terms of the nocebo effect, because Alzheimer's is a really scary disease. I mean, I'll take a heart attack any day of the week over going out with Alzheimer's. It's a very, very nasty disease, both for the person who's suffering from it, and even more so, arguably, for the people around them. And most people would choose just about any type of death, like great white shark attack. It's scary, but it's over pretty quickly, right? And so when people are told that it's like almost inevitable that they're going to get Alzheimer's, that creates some real fear. And that living in that fear state with your amygdala activated, and the downstream consequences of that in terms of like being in a constant fight or flight response, of course, can become a self-fulfilling prophecy, because being in that fight or flight response will exacerbate inflammation and oxidative stress if it's a chronic thing. So
Absolutely. Yeah.
I know, for a while it was like early on, that kind of information was only being delivered by a genetic counselor who could explain some of these things. But now, since consumers have access to it, my concern is just I'm glad to start seeing that we're starting to see some studies like the one in 2019 about the effects of genotype on exercise. But I hope we see more studies, even about like the effects of people knowing about some of their other SNPs and how that impacts their behavior and even their disease risk prospectively over time.
Yeah, and I think a lot of people are doing this, with good intentions, right? They want people to know about their risk, but I
Sure.
mean, the problem is that to date, there's very little evidence to suggest that telling somebody about their risk is going to change their behavior. So
Right.
if you were trying to tell ApoE4s that they're carriers because then they're more likely to focus on their sleep and meditate and exercise and eat a high quality diet, that would be great, but there's only a very small percentage of the population who are going to respond in that way, and the rest of them are then just going to spend a lifetime worrying about the fact that
Worrying.
they get
Yeah,
Alzheimer's.
exactly.
Yeah.
This is why I've become such a powerful believer in health coaching, because
Hmm.
information is not enough to change behavior. We
Absolutely,
know this
absolutely. Yeah.
beyond the shadow of the doubt. There's the famous CDC study that found that only 6% of Americans consistently engage in what they define as the top 5 health behaviors. So again, not we're not talking about intermittent fasting and keto and CrossFit and all of this. We're just talking about maintaining a healthy weight, getting enough exercise, not smoking, not drinking excessively, and getting enough sleep. They didn't even include diet anywhere in those 5 health behaviors. And yet only 6% of Americans engage in them. And I guarantee you, if you take, 100 people off the street and you say, is it healthy to drink a lot and smoke and not exercise and be overweight? They're going to say no. So they
Yeah.
know it. They
Mm.
they have the information. They just are not able to change their behavior. So for me, it's like, what if we took some of this huge amount of financial resources that are being invested in research on SNPs and all of this stuff, and put it towards like really action, a huge public health effort that involved scaling health coaching and learning more about how to actually assist people in changing their behavior and support them doing that over a lifetime. Like, imagine the impact that would have versus this.
Yeah. Which has been shown already to basically not make any difference. If you're trying to tell people about their risk, it doesn't change their
Right.
behavior. So
Right.
yeah.
So yeah, it's fascinating. I love that you have done this deep dive, and are out there sharing this information, because I mean, you know as well as I do that there's a lot of misunderstanding, and practitioners like me have contributed to that misunderstanding. So I consider this my act of public service to rectify that and help get the word out, and so that we stay current with what the research actually really is showing
Mm.
us. And I mean, this isn't the first time that we have misunderstood something in medicine. If you look back, I always like to say the history of medicine and science in general is most people being wrong about most
Yeah.
things most of the time. So the best we can do is correct those misunderstandings as we go, and I appreciate your efforts in doing that with the genetics and single nucleotide polymorphisms.
Thanks. I'm seeing as my day job is being an academic, I am very familiar with the concept of being wrong most of the time, and that's just part of the job. So maybe that sort of
Yeah, that's
helps.
what you sign up for as a
Yeah,
scientist, right?
exactly. Yeah.
So yeah, Tommy, where can people find out more about your work? I know you're involved in a lot of interesting stuff.
So the easiest thing at the moment is probably go to drragnar.com, drragnar.com. Ragnar's my middle name. That's my website, and it has
That's a
links
way
to.
cooler web URL than drwood.com.
Yeah, exactly. Drwood.com may have been taken as well when I started. But Yeah, so I have like podcasts on there, and there's some old blog posts. I haven't blogged in a while, but you go there, and you also have my CV and the papers I've published and stuff. And then Instagram at Dr. Tommy Wood. I usually post stuff on there, like when I publish a paper again or go on a podcast. And yeah, there's a couple of companies that I'm in the process of trying to work with to do exactly the things that you mentioned. Try and scale some of these health behaviors. Will help people support their behavior change, and as and when those become successful, then those mediums will show that. So those are the best places to go.
Yeah, it's interesting that a number of our colleagues have all kind of reached the same conclusion after being in this field for many years, which for some people will be good news and others will be bad news because when I reflect on what is behind this, the desire for or maybe what turns out to be the fantasy that genes are going to solve everything, is certainly it would be easier, right? If we could develop a kind of gene modification technology or even therapeutic drugs that would target certain genotypes, I wouldn't have to do anything. I could just keep living my same lifestyle and eating the same diet and not have to pay the price. So when it always keeps coming back to the simple things that we talk about, like cleaning up your diet, moving more, managing your stress, I think it's a double-edged sword, or it's good news, bad news for people, because change is hard. Let's face it. If change wasn't hard, we wouldn't have only 6% of people following those top 5 health behaviors. And I don't care who you are, change is hard. Some people are just more successful at it, but even for them, it's difficult. And so I'm really interested, with our launch of the health coach training program that's now just enrolled its 3rd cohort. That's what I'm investing in for the future, because I really believe that behavior change is the only way we're going to reverse the epidemic of chronic disease. I don't think fancy designer genetic drugs are going to do it. I don't think any drugs are going to do it. And I don't think just hammering people with information is going to do it either. So we've tried that. It doesn't work.
I completely agree. And I think you're right. It's a double-edged sword, because we're fairly certain that most of the things that we're doing in academia and medicine aren't really going to help that much, despite the huge amount of money that we're going to spend. But we probably do know what the answer is. It's just difficult. It's simple.
It's not that sexy, and
it's not sexy. It's simple, but it's not easy.
You can't patent it and make $1 billion off it either. So there's that problem as well.
We've both realized that, I think.
It's hard to raise venture capital on that, on getting people to stop eating pizza and donuts, right?
Yeah, definitely.
Yeah. All right, Tommy, thanks again for joining us. Appreciate your work, and love to have you back in the future after your next deep dive.
Likewise, Chris. Thank you so much for your time.
Take care. Bye-bye.
Bye.
That's the end of this episode of Revolution Health Radio. If you appreciate the show and want to help me create a healthier and happier world, please head over to iTunes and leave us a review. They really do make a difference. If you'd like to ask a question for me to answer on a future episode, you can do that at kriskresser.com/podcast question. You can also leave a suggestion for someone you'd like me to interview there. If you're on social media, you can follow me at twitter.com/kriskresser or facebook.com/kriskresser L.A.C. I post a lot of articles and research that I do throughout the week there that never makes it to the blog or podcast, so it's a great way to stay abreast of the latest developments. Thanks so much for listening. Talk to you next time.